A very good morning to all. In this video, we are going to study about the structure of nucleus and the structure of chromosomes. So let's start with the structure of nucleus. So as you know that nucleus is the important part of the cell which direct and control all cellular function. So the nucleus is also called the director of the cell or the brain of the cell. Why? Because it directs all cellular functions. जितने भी सेल के अंदर functions हो रहे हैं सबकी डायरेक्शन कौन प्रोवाइड कर रहा है? न्यूक्लियस प्रोवाइड कर रहा है. That's why nucleus is known as the director of the cell. So it is first described by Robert Brown in early 1831. Later on, the material of nucleus is stained by the basic dyes and was given the name chromatin by Fleming. फ्लेमिंग ने क्रोमेटिन जो कि मेन मटेरियल है न्यूक्लियस के अंदर जो प्रेजेंट है उसको डिस्क्राइब किया. Nucleus is absent in all prokaryotic cell. As you know that there is no membrane bound nucleus that is present in prokaryote. So the material, that is nuclear material, that is present inside the cytoplasm, scattered here and there in the prokaryotes, but it is present in eukaryotic cells. So again exceptions are there, and the most important exception is erythrocytes, that is RBC of many mammals, then sieve tube of phloem, tracheids and vessels of xylem. These are the permanent tissues of the plants. So the nucleus is absent in all these cells. So typically a nucleus consists of following structure, that is nuclear membrane, nucleolus and nucleoplast. As shown in the diagram, there is a central part, that is nucleolus, and above there is a nucleomembrane and some liquid material that is present, semi-liquid matrix is present over here, that is nucleoplast. So let's start with the first one, that is nuclear membrane, or it is also known as nuclear envelope. So it is a membrane that is double membrane and two parallel membrane with a space of about 10 to 50 nanometer, and the space is known as perinuclear space. As shown in the diagram, there are two layers that are parallel to each other. One is outer membrane and other is inner membrane, and a space showing here between these two membrane, that is perinuclear space. So what is the function of this perinuclear space? This perinuclear space forms a barrier between the material that is present inside the nucleus and that is present inside the cytoplasm. So क्या है ये पेरीन्यूक्लियर स्पेस? एक बैरियर, एक दीवार है एक तरह से, किसके बीच में? जो न्यूक्लियस मटेरियल आपका न्यूक्लियर के अंदर प्रेजेंट है और जो साइटोप्लाज्म है, साइटोप्लाज्म और इन दोनों के बीच में एक तरह से बैरियर है. So the outer membrane usually remain continuous होती है outer membrane with the endoplasmic reticulum, and it also bears some ribosomes on it that are the protein factory. The nuclear membrane is interrupted by minute pores which are formed by the fusion of its two membranes. Some pores are present in the nuclear membrane that are because of the fusion of inner and outer membrane. So this nuclear pores are specially made for the passage through which the movement of RNA and protein molecules takes place between the nucleus and the cytoplasm. Means exchange होता है यहाँ से nuclear pore से specially. क्या exchange है? किस चीज का exchange है? Exchange है RNA and proteins का. Normally there is only one nucleus per cells, but some variations are also there in which the number of nuclei are frequently observed. Now, what are the functions of nucleus? It allows the passage of inorganic ions, small organic molecules, ribosomal units, RNA and protein through nuclear pores, or you can say it control nucleocytoplasmic interaction and exchange. It help in the exchange between the nucleus and cytoplasmic interaction. It also help in the exchange of various components through nucleus and cytoplasm. It also maintain the shape of nucleus. So this is the structure of nuclear membrane and its function. The next component of nucleus is nucleoplast. As the name shows, it is plast, means it is a semi-fluid, or you can say liquid. So it is a transparent, homogeneous, semi-fluid that is colloidal in nature, and you can say it is the ground substance that is present in the nuclear membrane. So all the activities that takes place inside the nucleus is provided, or you can say it is completed in the environment provided by the nucleoplasm. It also helps in maintaining the shape of nucleus, so it acts as a nucleus skeleton. The nucleoplasm also contains two special features, that are nucleolus and chromatin. So the third part of nucleus is nucleolus, and it is a darkly stained spherical body found in nucleoplasm that is without any membrane and is composed of large amount of ribosomal protein and ribosomal RNA. It is generally associated with nucleolar organizer region, or you can call it NOR, of the nucleolar chromosomes. Means जो chromosomes specially एक portion है जहाँ से क्या है, there is a you can say formation of nucleolus takes place. Larger and more numerous nuclei are present in cells activity carrying out protein synthesis. So special जो आपके cells है जिनका main function आपका protein synthesis ही है, वहाँ पे आपको जो nucleolus है उसका size भी बड़ा मिलेगा और उसका count भी ज़्यादा होगा. Now, what are the function of this nucleolus? It is a seat for the biogenesis of ribosomal RNA and also stores ribosomal RNA. It is also play an important role in the spindle formation during the cell division. Now, the most important thing that is present in nucleus is chromatin fibers, or you can call them nuclear chromatin. So these are loose nucleoprotein fibers. These are thread-like structure that is present uniformly distributed in the nucleoplasm, but observed only when there is an interphase stage or during the cell division. This is the first stage, you can say, uh, of the cell division, where the condensation, or you can say the coiling of chromosomes takes place. Okay, so during cell division, the chromatin material undergoes condensation by coiling and dehydration of to form chromosomes. Uh, what is these chromatin material? Actually, these are the thread-like structure that are scattered over here and there in the nucleoplasm. क्रोमेटिन क्या है? थ्रेड फाइबर्स हैं जो कि आपके इंटरफेस में नजर आएंगे सिर्फ. उसके बाद जब सेल डिवीजन शुरू हो जाता है तो सेल डिवीजन के टाइम पे ये कंडेंस हो जाते हैं, इकट्ठे हो जाते हैं, कॉइल करते हैं एक दूसरे के साथ में और क्रोमोसोम्स की फॉर्मेशन करते हैं. The chromatin containing DNA and some basic protein called histones and non-histone proteins and RNA. As the diagram shown over here below, you can see that after condensation, the DNA formation takes place and DNA is coiled by some basic proteins, इनके ऊपर coiling हो जाती है proteins की, that are histone, because DNA is negatively charged and proton are the positively charged. तो यहाँ पे क्या होगा, उनकी कॉइलिंग हो जाएगी. कॉइलिंग के बाद में जो फॉर्मेशन होगी वो न्यूक्लियोसोम्स की बनती है, और बहुत सारे न्यूक्लियोसोम्स जब आपस में इंटरकनेक्टेड हो जाते हैं वो सुपर कॉइल्स बना देते हैं, और सुपर कॉइल से जो फॉर्मेशन होती है दैट इज द क्रोमोसोम, दैट लुक्स that you can say transfer the characteristics from one generation to another generation. Okay, this is clearly shown in the diagram. What that DNA किस फॉर्म में प्रेजेंट है, डीएनए जो है आपका क्रोमेटिन फॉर्म में प्रेजेंट है, जो लूज अनकॉइल्ड यहाँ वहां पड़ा हुआ है न्यूक्लियोप्लास्ट में, वो कॉइल होगा, कॉइलिंग होके कंडेंस होके डीएनए बनाएगा. डीएनए से क्या बनेगा, उसके ऊपर डीएनए नेगेटिव चार्ज है, उसके ऊपर पॉजिटिव चार्ज हिस्टोन्स की बाउंडिंग होगी, कुछ नॉन हिस्टोन प्रोटीन्स ऐड होंगे, न्यूक्लियोसोम्स की फॉर्मेशन होगी, सुपर कॉइलिंग होगी, एंड देन क्रोमोसोम फॉर्मेशन टेक्स प्लेस. So let's study the structure of chromosomes. As you know that in human being there are 23 pairs of chromosomes are present. It is different in different species. Okay, so the structure of chromosomes is because of the center, that is centromere. As shown in the diagram, there is a portion between the yellow lines, and this portion is the centromere region of the chromosomes. So it has a primary constriction that is known as centromere. इस सेंट्रोमियर के ऊपर कुछ डिस्क लाइक स्ट्रक्चर प्रेजेंट है, एंड दीज डिस्क लाइक स्ट्रक्चर आर नोन एज नाइटोकोर्स. ओके, यहाँ पे आपको येलो कलर्स के नाइटोकोर्स नजर आ रहे हैं, जिनके साथ में आपको बहुत सारे माइक्रोट्यूब्यूल्स नजर आ रहे हैं. ओके, सो यहाँ पे जो सिस्टर क्रोमेटिड्स हैं, आपको दो पार्ट्स नजर आ रहे हैं क्रोमोसोम के, टू पर्पल पार्ट्स आर देयर, जो कि आपस में क्या है, ज्वाइंट है. किसके कारण ज्वाइन है, बिकॉज ऑफ द सेंट्रोमेयर. सो दिस इज द बेसिक स्ट्रक्चर ऑफ अ क्रोमोसोम्स. ये जो नाइट्रोफोर लेयर बनी है, जो डिस्क बना है, ये आपके सेल डिवीजन में मेटाफेज के टाइम पे बनता है. ओके, सो दिस इज द बेसिक स्ट्रक्चर ऑफ अ सिंपल क्रोमोसोम that is having two sister chromatids that get joined with the help of centromere region. Now, based on the position of the centromere, the chromosomes can be classified into four types. What are these four types? These are metacentric, submetacentric, acrocentric, and telocentric. अभी क्या है structure? If the chromosome is metacentric, the last image, or you can say the image on your right hand side, यहाँ पे क्या है, आपका सेंट्रोमियर बिल्कुल सेंटर में प्रेजेंट है. वो क्या करेगा, इन क्रोमेटिड्स को इक्वल आर्म्स में डिवाइड करता है. फॉर एग्जांपल, ऊपर वाली आर्म को हम पी आर्म लेते हैं और नीचे वाली आर्म को क्यू आर्म लेते हैं, तो द साइज ऑफ पी एंड क्यू आर इक्वल. द सबमेटासेंट्रिक क्रोमोसोम, इन विच द लेंथ ऑफ द arms are different, one is shorter and one is longer. Okay, so क्या होगा, जो सेंट्रोमियर है वो एंड की तरफ मूव करना शुरू कर देगा. In acrocentric, the third from your left, or you can say the second diagram from your left hand side and third from the right hand side, it shows that the centromere is closely near to the end of the arms. So this type of structure is known as acrocentric structure. The last one, that is telocentric. So the telocentric has the terminal, or you can say at the end of the chromosomes, arms बिल्कुल आपके एंड में होगा. ओके, तो क्रोमोसोम के एंड में प्रेजेंट है. आपका फर्स्ट डायग्राम दैट शोज द टीलोसेंट्रिक टाइप ऑफ क्रोमोसोम्स. Sometimes few chromosomes have non-staining secondary constriction at some constant location, and these give an appearance of a small fragments known as satellite. Okay, so satellite region कहाँ है, आपके किसी भी एंड में, क्रोमोसोम के किसी भी एंड में कुछ ऐसी नॉन स्टेन्ड आपके लोकेशंस आ जाती है, जो कि ऐसा नजर आता है कि आपकी नई फ्रेगमेंट शुरू हो गई है क्रोमोसोम्स की. सच टाइप ऑफ क्रोमोसोम शोज सैटेलाइट regions, or you can say satellite constriction. So in this video we have studied about the structure and the function of nucleus and the structure of chromosomes and its function.